Hey everyone, and welcome. Have you ever wondered how scientists manage to keep human cells, the actual building blocks of you and me, alive and well in a petri dish, totally outside of the human body? It's one of the most fundamental things in modern medicine, and today we're going to pull back the curtain on the secret sauce that makes it all happen.
So let's just think about that for a second. If you treat a single cell like a tiny little person, what does it actually need to survive? I mean, it needs a complete artificial world. Everything from food and water to the right kind of air. The big question is, how on earth do you provide all of that?
Well, the answer is this amazing stuff called a cell culture medium. And honestly, the best way to think of it is as a life support system in a bottle. It's this super complex liquid cocktail of salts, sugars, vitamins, amino acids, the whole shebang, all perfectly measured out to mimic the environment inside our bodies and give those cells exactly what they need to not just survive, but to actually grow.
But, before we had all these high-tech, perfectly engineered formulas, where did scientists even start? Let's rewind the clock a bit and take a look at the very first attempts at nature's original recipe.
I mean, it totally makes sense, right? If you want to grow cells outside the body, your first thought is probably going to be, well, what are they used to inside the body? So yeah, the very first cell cultures used natural body fluids like plasma and lymph, literally the stuff that our cells are swimming in every single day.
And out of all these natural fluids, one ingredient quickly became the undisputed champion, serum. This kind of golden-colored liquid, which is just what's left behind after blood clots up, turned out to be the original magic ingredient. It's what really made long-term cell culture possible in the first place.
So, to get a little more specific, what is serum? Well, it's basically blood plasma, but with all the things that make blood clot, like fibrinogen, taken out. And by removing those, you're left with this incredibly stable, nutrient-packed cocktail of proteins and hormones that cells just go crazy for.
Okay, but here's where the story gets complicated. This magic ingredient, this amazing gift from nature, well, it turned out to be a classic double-edged sword. It worked, but it also came with a ton of really serious problems.
On the one hand, serum was incredible. It was like an all-you-can-eat buffet for cells, providing all the nutrients, hormones, and minerals they could want. It had growth factors that screamed, divide, divide, divide. It had proteins that helped cells stick to the dish, and it even acted as a buffer to keep the chemistry just right. It was the perfect all-in-one package.
But, and this is a big but, the downsides were huge. First off, serum is really expensive. And worse, its exact chemical makeup is a total mystery, a black box. It changes from one batch to the next, which means your experiments are frustratingly inconsistent. Plus, there was always a high risk of contamination. And if you're trying to make a pure drug using these cells, all that extra mystery protein in the serum is just a nightmare to get rid of. So, this huge problem, it actually sparked a revolution in biology. Scientists realized they had to move away from the mysterious alchemy of using serum and shift towards the precision of engineering. They set out to build the perfect cell food from scratch.
And this right here really shows you that massive shift in thinking. We went from using natural media, where the recipe was basically unknown, to creating artificial media. And with artificial media, every single ingredient is known, purified, and added in a precise amount. It's kind of like the difference between using an old folk remedy and using a modern, manufactured medicine.
The amazing thing is, today you can just buy these artificial media right off the shelf as standardized, quality-controlled products. A really famous one is called RPMI 1640. It's a totally defined recipe, which means scientists in New York, Tokyo, and London can all use the exact same formula and get results they can actually compare. It's a game changer.
Now, this evolution didn't happen overnight. It was a slow, deliberate process. It all started with just simple, balanced salt solutions, basically just salty water to keep cells from bursting. Then came basal media, like MEM, which added the essential building blocks like amino acids and vitamins. That led to more complex formulas like RPMI, and eventually, the holy grail, serum-free and chemically defined media, where that mysterious magic of serum was finally totally replaced with a known list of pure ingredients.
So the big takeaway here is that we've gone from relying on this one, single, undefined natural soup to having this massive catalog of specialized artificial media, each one custom-tailored for different kinds of cells and different kinds of experiments.
Okay, so we've got the perfect recipe, right? We're done? Nope, not even close. It turns out having the perfect food is only half the story.
You can give cells the best 5-star meal in the world, but they just won't thrive unless their physical environment is absolutely perfect. So, beyond what's actually in the bottle, there are a few golden rules, these non-negotiable physical and chemical conditions that scientists have to get right to keep their cells happy and healthy.
First up, temperature. For human cells and most other mammals, the sweet spot is 37 degrees Celsius. Sound familiar? Yep, it's our own body temperature. A little too hot and the proteins in the cells start to fall apart. A little too cold and their growth just grinds to a halt. It has to be just right.
Now this is super cool. How do you know if the cells are happy with the chemistry of their environment? Well, you have to watch the pH, the acidity. The perfect range is between 7.2 and 7.4. And to keep an eye on this, most media contain this amazing color indicator called phenol red. It acts like a little traffic light. When the pH is perfect, it's this nice, healthy reddish pink. But as the cells live and create waste products, the medium gets more acidic and turns yellow, a clear signal that it's time to give them some fresh food.
Next up, oxygen. Just like us, cells need to breathe to create energy. But here's the catch. It's a really delicate balance. They rely on the oxygen that's dissolved in the medium. But too much oxygen can actually be toxic. It creates these nasty things called free radicals that can damage the cell. So the oxygen level has to be carefully controlled.
And finally, there's something called osmolality. That's just a fancy word for the salt concentration. The key is that the medium has to be isotonic, which means it has to have the exact same salt balance as the fluid inside the cells. If it doesn't, water will either rush into the cells until they burst, or it'll get sucked out, causing them to shrivel up like a raisin.
So, when you step back and look at the whole picture, the journey of cell culture media is just incredible. We've gone from relying on a mysterious, unpredictable magic ingredient from nature to developing these precisely engineered formulas, all while learning how to perfectly control the tiny physical world of the cell. It's truly the quiet, unsung hero behind so many of the breakthroughs we see in medicine and biology.
And all of this leaves us with one last big question. As our ability to design these tiny life support systems gets more and more sophisticated, what's next? We've already moved beyond the need for serum in a lot of cases, so is it possible that one day we could create a medium so perfect, so advanced, that it allows us to grow entire, complex human organs right there in the lab? The story of this humble liquid suggests that the possibilities are really just beginning.